Again, okay. Thanks, Christina, for the introduction. It's my pleasure to be here. This is my first meeting, as Christina mentioned, and I could already feel the sorry. I'm happy to be here. Sorry, I'm taking a bit of time. Okay, so it's my pleasure to introduce Tina Reinis-Dottir. I'm good. Okay, yeah, I didn't butcher your name. She's a PhD student in the University of Iceland in Dr. Hans Jonsson's lab. She has been working on understanding the molecular mechanism of the protein KMT2A, and she has recently published a paper showing that how mutation in a particular region of this protein might have this broad spectrum of symptoms that we see in WSS. So without taking much of the time, I would like to welcome Tina and show her work to you guys. Thank you.
Thank you so much for the introduction. So I am Tina Reinis-Dottir, and I am a PhD student, like he said, and my PhD project is on researching Wiedemann-Steiner syndrome. So I'd like to, just before I begin, I'd like to just thank everyone for the opportunity to be here. It's so fantastic to be able to see the community with my own eyes. And yeah, I hope the conference will be as good as it's starting out to be. So my project is on establishing postnatal malleability of Wiedemann-Steiner syndrome, which is really important for us. So this is my presentation outline. So I want to give an introduction, then I want to move into describing how our mouse model functions. And then I want to spend the majority of the time talking about how the characterization of the mouse model is going. And then finally talk a little bit about our progress in postnatally knocking in KMT2A, which will all become a bit more clear once I get to it. So Wiedemann-Steiner syndrome is caused by heterozygous disease-caused invariants in the KMT2A gene, as has been said before. Now, KMT2A gene transcribes the protein KMT2A, which is my protein of interest in my research. And this protein is a very large multi-domain protein, and it is a lysine histone methyltransferase 2A, and that's where it derives its name from. And it is responsible for placing mono, di, and trimethylation marks on lysine 4 of histone 3. So it's in the epigenetic machinery. And what has been researched so far within this protein is that it plays a very critical role in hematopoietic stem cell differentiation as well as embryonic pattern development. And this protein is very interesting. There is supposed to be an image here, which does not appear on the slides. Oh, there it is. Okay. Yes, so this protein is very interesting to us because it is post-translationally cut into two protein products, which is the N and the C terminal. And then subsequently it heterodimerizes to form a functioning complex. And this is very interesting to us, and why this protein does this, we don't know. And this enzyme taspase 1, which is responsible for cutting it, doesn't have any other purpose that we know of, which is also very interesting. And before I move into the mouse model, like he said, we published a paper this June, which was my first paper, and we made some very interesting discoveries within the protein itself. So before I move on to the mice, I just want to give you a quick brief of what our sort of biggest finding was. So what we discovered is that, again, the image is maybe not up here. Oh, there it is. Okay. So what we did is that we compared all of the missense variants found in Wiedemann-Steiner syndrome and compared them to a control population. And missense variants are variants where only one amino acid in the whole protein is substituted for another amino acid. And what we found is that we found a very, very great enrichment of missense variants found within one specific domain, which is the CXXC domain. And the CXXC domain is responsible for binding unmethylated CpG sites at the DNA. And so it serves a DNA binding function. And so what is sort of the biggest draw from this one is that I guess it's what we didn't see. And what we didn't see is that we didn't see any enrichment of variance within the set domain, which is the second to last domain in this figure. And so on the x-axis are all the domains in order, and then on the y-axis is the percentage of observed variance. And the red dots represent Wiedemann-Steiner syndrome variance, while the gray dots represent control variance. And what you can see is that on the CXXC domain, we see a great spike in percentages within Wiedemann-Steiner syndrome patients. And we also saw a slight enrichment in 2 other domains, which is the PhD1 and PhD4 fingers, which confer stability to the protein. And the biggest thing that we didn't see is that we didn't see an enrichment in the set domain. Now, the set domain holds the enzymatic activity, and so far our hypotheses have been sort of circling around, since this function or this protein is mostly known for its enzymatic function, we sort of drew the conclusion that the enzymatic dysfunction would be the mechanistic underlaying for this syndrome. However, with this finding, we're sort of shifting our focus away from the set domain, although it is very important, but we're sort of moving our focus maybe into the unexplored DNA binding sort of aspects of the protein. So I think making this discovery such early on, especially within my research, is going to be very useful and is going to open up a lot of other windows of opportunities to sort of look into. And what we also did is that we looked at other epigenetic regulators to see whether this was specific to KMT2A or whether the CXXC domain is just that important. So if you have variants there, it just is going to lead to disease. And what we saw is that it seems to be specific to Wiedemann-Steiner syndrome. So this is sort of the biggest finding, but this paper is online if you care to look into it a bit further. So what we want to also, like the big question, I guess, is what the window of opportunity for genetic diseases is. And these sort of are the big questions that I want you to keep in mind throughout this presentation. So of course, there has been substantial progress made in sort of genetic research and to help genetic disorders make it into clinical trials. But it is not always clear what the window of opportunity is, and to sort of know at what time the syndromes can be potentially treated. So we wonder whether if we could rescue the gene expression postnatally or in utero, whether that could if we could do that, then I guess we could ask the question, is Wiedemann-Steiner syndrome treatable during pregnancy or after birth? So that's sort of our big goals here. So yeah, next I wanted to go into and spend some time on describing how our mouse model function and why it is a novel model, and all of the details regarding that. So our mouse model has a heterozygous cassette insertion in intron 1 of KMT2A. Now, what that means is that we have a cassette that's inserted very early on in the gene, right at the front, only affecting one allele. And this cassette is flanked by LOXP sites on either side. So that would result in a premature termination of the mRNA transcript. So that's so what it means is that we have a LOXP site, the cassette, and then a LOXP site. And that's also where we derive the nomenclature from. That's why we call these mice KMT2A lock-stop LOX or KMT2A LSL. And that's how we refer to this model. And what this means is that by inserting this cassette in this place, it contains a terminating sequence which results to the premature stop of the transcript. And this would lead to a heterozygous expression of KMT2A, so only the unaffected allele makes a transcript which is translated into protein, and this would then lead to Wiedemann-Steiner syndrome. So I just wanted to describe a little bit better how the concept works. So by placing these LOXP sites on either side of the cassette, we're not only creating a knockout, we're also utilizing the Cre-LOX recombination system to be able to postnatally remove the cassette. So we have we were able to manipulate this and have a temporal control of when we knock in the full expression, when we remove the stop codon. And to do this, we breed our mice to a Cre-containing mouse model, and then we treat the offsprings with tamoxifen. And what that does is that the cytoplasmic Cre recombinase is released into the nucleus, and then it cuts at the LOX-P sites to recombine the cassette. All right, there we go. And this gives us good temporal control and good regulation of when we want to try to knock in the protein and see essentially what happens. And in the big scheme of things, this is all to see if we can rescue KMT2A expression, will that rescue any Wiedemann-Steiner syndrome phenotypes? So this is sort of how the mechanistic basis of it. Of course, it's I guess a bit difficult technically, but this is sort of the general idea and how and why we created this model. And this has never been done before and is a great and a very essential reagent for my research and of course for the community. So next up, I just wanted to spend some time and talk a little bit in detail how we're characterizing these mice and what sort of phenotypes they're showing. And I know this is probably what you've all been waiting for a little bit, but so we've done some preliminary phenotyping of the mouse model so far, and which has been conducted on relatively small cohorts. And like I said, here we're really trying to establish which phenotypes this mouse model displays, which are relevant to Wiedemann-Steiner syndrome. And we really want to see whether we can establish very robust phenotypes, which can be then used as metrics for our rescue. So first off, for what we've just seen by simply looking at these mice is that they have a fur phenotype. We're not sure how we're going to put the definite metric to it, but it's definitely the fur is grayer and is a bit more ruffled in a sense. And we're really hoping to be able to use some good tools to describe this in more detail, but we find this very interesting that an animal which is furry has a fur phenotype. I guess that was a big surprise to us and has never been described before. And on these images, you can see two siblings, so the wild type being on the left and the KMT2A mice on the right. Another phenotype that we've seen is the shape of the skull. We see that the snout of the mouse seems a bit pushed back and a little bit shorter, and then the skull seems a bit more arched. And this is one of these phenotypes that we've just seen, and we are hoping to get a good MRI scan that's coming hopefully to our facility soon to be able to take some x-rays and be able to look at this more definitely. But this is very interesting and very stands out to us. And we're also looking forward to looking at other places. We're also looking at the hips and other regions, but this is so far what we've just seen by simply looking at them. Now, for a more definitive phenotype, our mouse model demonstrates growth deficiency, and we see this both in our males as well as our females. And in these graphs, you can see the weight of the mice from 2 to 10 weeks. And again, the wild type mice are here in gray and the KMT2A lox mice are in blue. And you can see that there's a significant weight difference. And on these pictures are siblings which are born on the same time from the same mom, but you can definitely see which one has the cassette. And this is something that stood out from us like right from the get-go and was very interesting. And there have been created models with a knockout before, and that has also been a phenotype which has been described. So this is very reassuring also that our model is functioning as it should be. Another thing that we've seen so far is that our mice demonstrate hypotonia. And I performed 2 strength tests on these mice at postnatal day 6, which is comparable to around a 6-week-old baby. And the first test is a surface riding test, and it tests their core strength, trunk control, as well as their coordination. And that's when I place the pups on their backs and record the time for them to flip over to their prone position where they sort of like to be. And so far we've seen is that our mice demonstrate increased surface riding time, so they're taking longer to flip back. And we do this in 3 trials consecutively, and we see that they repeatedly take longer to flip back. So this might be a good indication of hypotonia. And then the second test that we've done is a hind limb suspension test. And this sort of is directly testing their hind limb strength. And it doesn't seem that just their hind limb strength is the sort of core reason for this. However, we do this also in 3 trials, and you can see sort of at the 3rd trial, they're significantly more tired, so they take a longer time to get back. So we're also looking to phenotype adult KMT2A mice. And I've done very, very limited testing on some cohorts, but so far I've been focusing on the Y maze, and the Y maze tests hippocampal dependent memory deficiency without relying heavily on motor phenotypes. And we want to stay clear of any very motor efficient heavy sort of test, because we want to be sure that what we're testing is not just their motor capabilities, because we have other tests for that, and we want to be sure that the results that we're getting is not just being overshadowed by any motor phenotypes. So for the Y maze, we place the mice in an open Y maze, and we let them just explore freely for around 8 minutes. And we are sort of relying on the mouse sort of initiative to explore. And what these mice sort of tend to do is that they tend to enter the arm that has been least explored. So what they tend to do is that they sort of go around consecutively, and that's when what we record as alterations or spontaneous alterations. And that's when the mouse has entered each arm consecutively, no matter which order. And we usually see this very high in our controls, but what could be an indication of memory dependent memory deficiency is that when the mice sort of keep re-entering the same arm that they came from. And so far, at least, we've seen a decrease in our mice for their total alterations, which you can see on the graph here. I guess, right here. So the wild type mice have a higher total of alterations, but when we correct them to the total arm entries that they make, we're not sure whether this is significant yet. Is at least looking very promising, but I'll need to repeat this many more times and have more mice to be able to tell indefinitely. But this so far is very promising as well. So now I just want to give you sort of the brief of where we're at with the postnatal knock-in of KMT2A. And like I said before, our goal here is to induce the recombination of the cassette, so being able to remove it from its genome postnatally and rescue KMT2A expression. And then, of course, our long-term goal here is to observe whether we can see any rescue of these phenotypes that I've detailed before. And of course, we're also looking at many others that I didn't really have the time to talk about today. But this is currently being optimized and is proving to be technically quite difficult. But I still wanted to show you how the data is looking right now. So if you look on the graph here, the bar in pink are our control mice, and these show around 100% expression. So this is just relative expression, and as they should be. And these mice were just treated with the vehicle, so not with the tamoxifen drug, which is vehicled in corn oil. And then the green bar here is the mice that were just treated with the vehicle as well. So these were not treated with the drug. And they show the expected around 50% KMT2A expression. And the bar in the middle in the blue is our mice that were treated with the drug. And this is just a graph from one of the many optimizations that I've been conducting, but the data is pretty much all looking like this. So without having to go into every detail of the consecutive optimizations that I've done, you can see here that there are definitely a couple of mice that seem to be responding to our treatment, and they seem to be responding to the tamoxifen treatment and regaining KMT2A expression. However, there are the majority of the mice that seem either not to be responding or maybe responding and just not showing increased expression. So there are definitely still some caveats that we're working on, and we're trying to get through this hurdle. So the big caveats is that we are not seeing complete rescue, or we're not seeing rescue in every animal. And we're really trying to work hard on getting the drug administered properly or better. And that's sort of what we're trying to do. And this is also one of the things that we have just discussed, is that it could potentially be due to a weak promoter on our Cree model, so not on our KMT2A model, but the mice that we breed them to be able to reduce this combination. So we've been sort of discussing and taking very seriously whether we should look into getting a new cream model, which could help us get some results faster. And we've also been discussing using a constitutional cream model, which means that we don't have to make the induction ourselves using tamoxifen or any other drugs, as that is proving to be technically very difficult. So in this manner, the CRE is just knocked in at embryonic day 11. So this is an embryonic stage. So this is sort of around mid-gestation. And I think with this model, we could it could help us answer the question both of whether Wiedemann standard is treatable during pregnancy, but this could also help us sort of sift through which phenotypes of the models are CNS or due to defects in the central nervous system, and which ones are not. So we're looking into using this CRE model while still trying to optimize and get the tamoxifen induction up and going. But in the meantime, we're also looking at getting a new CRE model, which could help sort of accelerate our research even faster. So just a summary for my presentation today. So we have done some preliminary characterization of the mice, and we do see Wiedemann-Steiner syndrome relevant phenotypes, which just in of itself is just amazing. And it really proves our point that this model is a good model for Wiedemann-Steiner syndrome. This is a good disease model. And I think that's the first step. And seeing that is just absolutely amazing. So far, we've seen some fur and craniofacial phenotypes, growth deficiency, hypotonia, and I'm also looking into establishing whether there is definitely some hippocampal memory deficiency, and that's sort of the experiments that I've been focusing on. And what I've also been doing, which I didn't show here, but I've also been culturing cells from our mouse model and using them in culture and trying to see whether we can get even more sort of molecular backgrounds using this model by culturing their cells and seeing how they behave. And yes, so the optimization of the tamoxifen-induced recombination is being conducted as of right now. And sort of the two biggest points that I really hope I'm getting across here is the cassette function has been validated. It can be removed and it can regain KMT2A expression. How we're going to get it to just reach its full potential, we're still working on, and I'm definitely putting my work into trying to get that optimized. But this is just such a big opportunity. So I feel like everything is going according to plan. Of course, COVID was a little bit of a setback to our sort of Project Outline plan, but everything is working pretty good. So just to end my talk, I wanted to, of course, thank the Wiedemann-Steiner Syndrome Foundation for letting me talk here and giving me the opportunity to meet with the community, as well as Dr. Björnsen, who is not here today, who is my PI and leading this project, as well as the Eimskip University Fund and my great colleagues, Leandros and Kimberly, who worked hard with me on my paper, as well as all of our labs. So thank you. I'll be happy to take any questions.
Thanks, Tina. Excellent work. I would like to open this for general discussion. If anyone has any questions, I can come to them.
Yes. Do you have any plans to do any research involving like bone density in the mice?
Bone density?
Yeah.
So in my lab, there has been done a lot of work in a related syndrome, which has been looking at sort of the differentiation and how sort of the bones are growing. I have not done so myself. It is not on my current plan, but I think for this model, there are so many unexplored opportunities. And I think down the line, it will definitely be done. I think it will be very interesting to see whether we could see any difference there. But as of right now, it is not on my plan, but I think it could be very useful to look into.
Do you have any speculation as to the KMT gene re-expression or what getting it back to normal, on if that's going to be homogeneous to everybody who is affected a little bit and those who are affected a lot? Is there any speculation on how that's going to appear when you're finished?
So, I mean, the model that we have is sort of mimicking a loss of function or a complete loss of the one allele. So this is not directly translatable to a missense variant, but I do, however, think that a lot of the missense variants are sort of leading to defects in that part of the protein. So be able to rescue the expression, I think, would be very applicable to at least the majority of the patients.
Would you be able to give us a better understanding of what the cassette is and how you developed it before you introduce it?
Sorry, could you repeat the question? I didn't quite hear you.
Could you give us a better idea of what the cassette is and how you developed it and introduce it?
Yes. So maybe if I just go quickly back a slide, I guess. Around here, maybe. So the cassette contains a terminating sequence. And what that means is that the sequence within the cassette itself induces a poly-A tail to the transcript, which is being transcribed. So it's in the first intron. So when the transcription starts, it starts at the first exon. And once it gets down to the first intron, what's introduced really early on, just right at the beginning, is a poly-A tail, which then leads that the transcript is immediately degraded. So it's just that as soon as it starts transcribing, it's degraded immediately. So this should result to no transcript from that allele. So the only unaffected allele is the one that's producing KMT2A. Is that answering your question? Hi.
Hi, thank you for being here. Could you just give us an overall sense of what the hippocampus does and more specifically, how it's affected by the KMT2A?
Yes, it was a great question. So the hippocampus is sort of what we've taken from other literature, and sort of why we're focusing on the hippocampus is based on previous work that has been done in syndromes such as Kabuki syndrome, as well as Rett and Rubinstein-Taybi, which are sort of related disorders. So of course, KMT2A, we don't know what it does in the brain, to be honest. But we really want to see what it does and where it mostly is. And I know it's very well expressed in the hippocampus. So that's sort of what we're focusing on. And the hippocampus is sort of responsible for a lot of like memory as well as cognitive behavior. So we're sort of taking more focus on that instead of other regions, which may be more into like motor capabilities or movements and such. So we're targeting the hippocampus to be able to try to assess the intellectual disability aspect of our model.
I had a quick question from someone
Yes.
online, and then we'll get to you, Jackie. The drug that's administered, is that a
Yes.
one-time or does it have to be sustained?
So that's an excellent question. So like I said, I have been trying to get this optimized for a little over a year now, and we've tried multiple methods. This drug is used to reduce or induce recombination for the Cre-Lox recombination system in a lot of other models for a lot of other purposes. And what tends to happen is that people go for what works for their model. Unfortunately, I haven't known sort of what works right now, but I've tried both single doses, consecutive doses for up to 5 days. So I've tried a multiple different sort of both frequency, dosages, administration methods, giving it to the moms through the breast milk, giving it straight to the mice. So I've gone sort of a couple of different routes, and we've also looked into using, and we have tried using the metabolite, which is sort of the drug is in its purest form. So we've also been trying to get higher dosages of tamoxifen administered without, of course, having sort of impact on the mice or their behavior. So it's a delicate line, and I'm really trying to figure out what works best without overdosing or giving too much of the drug. But it could be that it needs to be sustained. So we're also looking into giving it earlier on in their age, and then also sort of continuously administering it throughout sort of their early life.
I think that tamoxifen in humans is used to treat breast cancer.
Yes.
So how did we come up with using tamoxifen in mice for WSS?
Yeah, that's a great question. So these mice that have the Cree model that we're using is sort of the key to this question. And this mouse model has, so what they have is that they have a fusion protein of the Cree and the estrogen receptor ERT2. And there's a fusion of these protein, and this does not affect the mice in any way. But what this does is that it retains the Cree in the cytoplasm. But when we administer tamoxifen, it binds directly to this fusion protein, and it allows the protein to enter the nucleus. And in that way, it can access the LOX-P sites, and it recognizes the LOX-P site, and that's where it cuts. And this Cree-LOX recombination system was actually discovered in bacteria, but we're using it in sort of genetics to manipulate a system, so where we can sort of exactly cut at a precise position to reduce any type of sort of genetic manipulation.
I was wondering if you could talk a little bit about, besides the way in which you've used this model so far,
Mhm.
what other ways do you anticipate this tool being used in the context of the entire research team?
That's an excellent question. So this model is like the pros of having a new model is, of course, that there's just unexplored angles everywhere. And even though I'm just doing a PhD, I really want to do everything. But I think that for like having such a powerful reagent is not just going to be a disease model, which is essential for these communities and for research to excel, especially for rare genetic disorders. But I think it's also going to be able to use, especially for other cell cultures, and being able to culture either, you know, we can, there's sort of just endless possibilities. We can look into pretty much anything. I'm looking at stem cells, we can look at neurons, we can look at sort of histological stuff, and I mean, I want to sort of go into the immune system, and of course we know that this protein has a lot of function in their stem cell production. So I think having created this model is going to be so valuable, and getting cells up and going, and in the cell sort of plate, we can also explore whether the recombination rescues any cell phenotypes. And that's sort of also what I'm doing sort of parallel to my mouse work, is looking at whether we can see the cells behaving differently and whether we can rescue the cell phenotypes, because that in essence is going to give us a hint whether this is going to translate to our mouse models. So that's also what I've been working on, is trying to get good results from the cells and able to sort of translate that also to a whole full body organism.
Yeah, and just to extend this conversation a bit, like, you know, this mouse gives us an excellent opportunity to tease out all the organs and look into them individually. Like this protein is also known to, you know, affect the generation of intestinal stem cells. And we know that most of our kids are affected with like constipation and all this kind of stuff. So it will allow us to, yeah, do many stuff apart from, yeah. If there are no more.
I have a question.
Yes.
Tina. So, you know, as you're researching from the mouse model perspective, and you heard our presentation earlier as we're setting in place these different tools, what does that normally look like as far as kind of the data that's collected from this community, say in Rare X, and what the characteristics you're finding in the mice, and how will that eventually match up, and how important is that?
I think it's going to be super important to get patient data. I think that's very, very valuable, both to us as researchers. And just looking at the first slide that I showed you, we would not been able to see that the CXXC domain seems to be having and playing a very critical role, and the DNA binding prospect of the protein seems to be relevant to this syndrome, if not for patient information and for people sharing their sort of results and just what they've seen in their kids. So I think it's going to be very important to get more of that. And of course, down the line, we would love to do a genotype phenotype. I know that's something that everyone is really interested in. And we would love to be able to explore whether we would see a better rescue, of whether we could see maybe different phenotypes within different domains or different types of mutation, and that's something that we're really interested in seeing. So if I'm thinking very broadly, I would love to explore, especially within my cell, maybe in mice eventually, whether just rescuing certain domains would be enough, because a limiting factor, unfortunately, to this is that KMT2A is very big, and for gene strategies, it's going to be a challenge getting KMT2A to get sort of higher expression within patients. So if we could sort of shift out which domains seem to be the important part, we could also help us accelerate faster into getting into gene therapy or other strategies. So I'd also love to use some CRISPR and try to just regain expression with just looking at different domains. So I think that will also sort of help us piece together this puzzle. And I think having a mouse model is sort of the biggest step into moving into patients. This is sort of the biggest link between sort of the molecular background and into therapeutics.
Tina, I have two questions.
Yes.
Like, have you looked into the myelination in this mouse?
I have not looked into myelination yet, no.
Okay, do you think that might be a problem? Like in the mice, it can be reduced, or it could be?
I mean, I would love to
Yeah,
look into that. I'm also on my next episode, I'm going to look at sort of just the structure of the brain. It has been seen in other diseases sort of relevant that some structural differences in the brain is also affected. So I'd love to explore whether that's something that we see in our model. I have not specifically looked at the myelin, but that would be a very interesting point to look at.
And have you done any aging studies in this mice?
The aging?
Yeah.
So far, we've not seen any decreased life expectancy or lifespan within our model. So these mice live just fine.
Okay, thanks. Oh,
Good morning tonight. I just
Hi.
had one question to kind of go off of what Jackie asked about the tamoxifen.
Yes.
For translating that to human use, would that actually be tamoxifen, or is that kind of a base to develop a new drug?
Yeah, that's a very good point. So the tamoxifen induction is used to manipulate the Cre-Lox recombination system. And in our patients, we do not have these LOXP sites. This is how we designed the model to be able to temporarily knock in the sort of expression. So the tamoxifen is used in combination with our Cre model. And by using the tamoxifen, which is not sort of administered as a therapy itself, it's used to induce the knockout of the cassette and sort of regaining expression. So this is more a genetic tool that we're using to see whether we're trying to answer the question whether KMT2A expression increase leads to rescue phenotypes. I guess that's sort of our biggest questions that we have, and sort of exactly my PhD project. But using tamoxifen as treatment is not going to be relevant for patients, since we're not utilizing the recombination Cre-Lox system in our patients.
Good morning. I'm sorry to have come in late. I wasn't here at the beginning of the lecture,
Of course.
but I just want to find out how far is this? I know this is still a preliminary or the beginning stage of this experiment, but how far can this, how far away can this be translated in when you start experimenting this on the kids or the humans?
That's an excellent question. So I mean, I am not a medical doctor, so I'm not really deep within the medical field and how this is going to be translated. So I guess I'm maybe not the person to be able to answer that question very accurately, but I would hope that our research, especially just within the progress within the last 2 years, is exponential. And designing a model, having a model, just seeing phenotypes that are relevant to this syndrome and knowing that this model is a good disease model has just propelled and hopefully shortened that timeline by a lot. But unfortunately, I don't know how long until this could be translated into gene therapy. But also hoping with other options of shortening the protein, or sort of looking more into what molecular basis is behind the protein, which is going to help us move even faster.
Okay, with no more questions, I'd like to thank Tina for the wonderful talk and wish you best of luck for your, next piece of studies.
Thank you. And I just wanted to say that I'll be here for the full conference. So if you have any more questions, feel free to approach me and talk about anything.
Thank you, everybody. And we're going to be taking a short break. We will start up again at 10:45 Eastern. 10:45 Eastern, we're going to go live again. So please be back before 10:45.